Now we're going to learn about the Calvin cycle. The Calvin cycle is called a non-light requiring reaction because, unlike photosynthesis, it doesn't require light energy to function. It takes place in the stroma of the chloroplast. The point of the Calvin cycle is to take the carbon from carbon dioxide and convert it into sugar. There are three phases of the cycle: carbon fixation, reduction, and regeneration. The first phase is called carbon fixation. It got its name because this is where the carbon, entering the cycle as carbon dioxide, is fixed or attached to a molecule named ruby P. Three carbon dioxide molecules enter the cycle one at a time, where an enzyme called rubisco attaches each carbon atom to a 5-carbon molecule called ribulose bisphosphate, or ruby P. The resulting 6-carbon molecule is an intermediate that barely exists for a millisecond before splitting into phosphoglycerate. Fun fact, rubisco, the enzyme that makes this happen, is the most common enzyme on Earth. As you can see here, the short-lived 6-carbon intermediate is converted into a 3-carbon phosphoglycerate. The 3-carbon phosphoglycerate is converted into another 3-carbon molecule called 1,3-bisphosphoglycerate. 6 ATP are used for this reaction. The next phase of the cycle is called carbon reduction. We start off with the 3-carbon molecule 1,3-bisphosphoglycerate. First, it is reduced by NADPH, which is converted into NADP+. Then a phosphate group is removed. The final product of this step is a 3-carbon molecule called glyceraldehyde 3-phosphate, or G3P. We have 6 of these, and one leaves the cycle to be converted into sugars elsewhere. So, now that one G3P has left the cycle, we are left with 5 G3Ps still in the cycle. The third phase of the Calvin cycle is called regeneration of the CO2 acceptor. As you may remember from the very beginning, the molecule that accepts the carbon as it first enters the cycle is a 5-carbon molecule called ruby P. So in this third phase of the cycle, ruby P is regenerated. First, 3 ATP are used in this process, and they turn into ADP. So think about it. At the beginning of this phase, we had 5 G3P molecules, each of which had 3 carbon atoms. That means 15 carbons. These are rearranged to form 3 molecules with 5 carbons each. And this new 5-carbon molecule is none other than ruby P. And here the cycle begins again. Ruby P comes into contact with the enzyme rubisco, which attaches it to 3 more carbon dioxides which enter the cycle. And now you've learned about the Calvin cycle. Congratulations!